Bismillah ar-Rahman ar-Rahim. Today we will discuss disaccharides and the beginning of polysaccharides. Okay. The completion of monosaccharides, you have to know them by name, enumeration only, because in the enumeration you will be asked in the exam about the classification of monosaccharides. Does it belong to which type of monosaccharide? Is it aldose? Is it ketose? Is it triose? And there are monosaccharide derivatives. You have to know the classification because in the exam you will get a name, a nominated example of monosaccharide, and they ask you about the classification. From these derivatives we have sugar. I will make a pen, a marker around the name of acids. We will discuss the formula, but the formula is not required from you. You don't need to memorize the formula. The example, you have to study them by name and to memorize them. Okay? The first example, aldonic acid, and there are examples, gluconic acid from glucose, galactonic acid from galactose. These are aldonic acids. The second is uronic acid. The first example is glucuronic acid from glucose, galacturonic acid from galactose. The third example is aldaric acids, such as glucaric acid. Ascorbic acid, it is vitamin C. It is a derivative of one type of the sugar, but it's not required from you to know from which sugar vitamin C is derived. Vitamin C has two names, either ascorbic acid or vitamin C. The second type of the derivative is sugar alcohol. The first is sorbitol, derived from glucose. The second is mannitol, derived from mannose. The third is dulcitol, derived from galactose. I'm going to discuss the formula in order to understand, but you need only to know enumeration, each type of the sugar or monosaccharide derivative belong to which classification. Is it sugar acid? Is it sugar alcohol? Is it amino sugar? To which classification only. This is an explanation by formula. If you remember from the first lecture, we have discussed this group. This group is an aldehyde group, which specify all aldoses. If we add 2 hydrogen atoms to this aldehyde group, we get alcohol, CH2OH. This is an alcoholic group, chemical group, or functional group named primary alcohol group. So aldehyde group CHO. If we add 2 hydrogen atoms, you will get primary alcohol group. If you add 2 hydrogen atoms to glucose, 2 hydrogen atoms to glucose, you will get sorbitol. If you add 2 hydrogen atoms to mannose, you will get mannitol. If you add 2 hydrogen atoms to galactose, you will get dulcitol. If you add 2 hydrogen atoms to fructose, you will get 2 types of alcohol, both sorbitol and mannitol. If you need to know why fructose gives sorbitol and mannitol, you have to study the formula. But only in the exam, you are required to know which are the sugars, alcohol, which are derived from fructose. Which alcohol does fructose give? Fructose gives sorbitol and mannitol. What are the functions or the biological functions of these alcohols in our body? The first example is glycerol. Glycerol is derived from glyceraldehyde. If you have studied the first lecture, we have said that glyceraldehyde is an aldose sugar. It contains 3 carbon atoms. it is an aldotriose. If we add 2 hydrogen, this is mean reduction, reducing. The glyceraldehyde will give us glycerol. Glycerol enter in the structure of triglycerides. What are triglycerides? These are the neutral fat, the lipids found in our body, and of course lipids has many biological functions. Glycerol enter in the structure of most phospholipids. We are going to study phospholipids in detail in the chapter of lipid chemistry. Ribitol is derived from ribose. We have studied ribose in the first lecture. Ribose contain aldehyde group. It is an aldose. Ribose contain 5 carbon atoms. It is an aldopentose. If we add 2 hydrogen atom to ribose, we get ribitol. Ribitol enter in the structure of vitamin B2, which has another name. Vitamin B2 has another name, riboflavin. Myoinositol. Okay? The structure, you don't know to need the structure or the formula of myoinositol, but myoinositol is considered sugar alcohol. If you look at the formula, it contains several alcoholic groups. It enters in the structure of one type of phospholipid termed lipositol. It has another name, phosphatidyl inositol. Of course, phospholipid has many biological functions. Very important to know the functions of phospholipid. We will study them later on in the chapter of lipid chemistry, and this is very important in the exam, the functions of phospholipids. The fourth type of derivative is the oxysugar. The oxy means we remove oxygen atom from carbon number 2. If you look at the formula of any monosaccharide such as glucose, you will find that carbon number 2 contain hydroxyl group. Hydroxyl contains oxygen. If you remove oxygen from carbon number 2, you will get the oxysugar, such as the oxyribose, which enter in the structure of DNA. L-fucose. 6. The oxy-L-galactose. This is an isomer of galactose. If we remove oxygen from the 6 carbon, you will get fucose. Fucose enter in the structure of blood group antigens, enter in the structure of glycoproteins. Of course, all these derivatives, you have to know the name, to memorize the name, and to know which classification the sugar belong to it. Is it sugar alcohol? Is it sugar acid? Is it deoxysugar? Is it amino sugar? So you have to know which type of derivative. And also you have to know in the monosaccharide, is it aldose? Is it ketose? And which type of aldose or ketose? Is it triose, 3 carbon atom? Is it belong to tetroses, 4 carbon atom, or 5, or 6, or 7 carbon atom? So all these chapter is enumeration, are about enumeration, and to know the example, to know the classification, and the biological function of certain types of monosaccharides. These are the formula. I will discuss it, but you are not required to know the formula. If you revise glucose, you will find the 6 carbon atom contain alcohol, but this is not an alcohol. This is carboxyl group. COOH is a carboxyl group, which specifies organic acid. Any organic acid should contain carboxyl group. So in glucose, if we oxidize the alcohol group at carbon number 6, it will be replaced by carboxyl group, and it will give us glucuronic acid. So glucuronic acid is an acid derived from glucose. This is another type of acid called gluconic acid. Gluconic, we oxidize the aldehyde group at carbon number 1. Glucaric acid from glucose also, we oxidize carbon number 6. The alcohol group is converted to carboxyl, and we oxidize the aldehyde group at carbon number 1. Ascorbic acid, you don't need to know the formula, and you don't need from which sugar ascorbic acid is derived, but you should memorize that ascorbic acid is a monosaccharide derivative. Ascorbic acid, that is vitamin C, is monosaccharide derivative. It is sugar acid. These are the first type of derivatives, which are sugar acids. The second type are sugar alcohol. If we get fructose and we add 2 hydrogen atom to fructose, fructose give us 2 types of sugar. The first is sorbitol. Sorbitol is the alcohol of glucose. From which type I know this is an alcohol of glucose? Carbon number 3 contain hydroxyl group to the left. And then glucose, there is an aldehyde group at carbon number 1. It is replaced by an alcoholic group due to reduction, addition of 2 hydrogen atoms. Mannitol, if we revise mannose, in mannose we have hydroxyl at carbon number 2 to the left, at carbon number 3 to the left, and also the aldehyde group is replaced by alcoholic group due to reduction. So fructose give us 2 types of alcohol: sorbitol, which is the sugar of glucose, alcohol of glucose, and mannitol, which is the alcohol of mannose. The fifth type of derivatives are aminosugars. Amino means amino group. Amino group, it is NH2. I will make a marking by the red pen around NH2. It is an amino group. Hydroxyl group at carbon number 2 of any sugar is replaced by amino group. That's mean we remove hydroxyl group at carbon number 2 and then we replace it by amino group, so we get amino sugar. Amino sugar, examples of amino sugar: glucosamine, which is amino sugar of glucose; galactosamine, amino sugar of galactose; mannosamine, amino sugar of mannose. And you have to know the abbreviation of the name: GLUN, GLU, abbreviation of glucose; N, that's mean amino group; galactose, and the abbreviation of amino group N; man, abbreviation of mannose, and the amino group N. Amino sugar enter in the structure of GAGs. We are going to study GAGs in polysaccharide, glycosaminoglycans. It has many biological functions. We are going to study glycolipids, glycoproteins in the chapter of lipids and proteins. Amino sugar enter in the structure of glycolipids, enter in the structure of glycoproteins. Of course, we are going to study the functions of glycolipids and glycoproteins, as well as the function of GAGs or glycosaminoglycans. Amino sugar acid, there is an extra acid added to amino sugar. You need only to know the name of this amino sugar acid. This is not an amino sugar alone. This is amino sugar with an added acid, organic acid. The example is NANA. You have to know the abbreviation because the abbreviation come in the exam. NANA, the abbreviation of N-acetyl. N means nitrogen. Acetyl, capital A, acetic acid. N comes from this N. Acetyl, we get capital A. Okay? Neuraminic, we get capital N. Acid, we give capital A. So you have to know from which the abbreviation is derived. This is a neuraminic acid. And we are going to see the formula of neuraminic acid, but of course, you don't need to know the formula. You need only to know the name of N-acetyl neuraminic acid and the classification. It belongs to which type of monosaccharide derivative? It belongs to amino sugar acids. The last one is ester. We are going to see them in the formula. This is neuraminic acid. If you look at the neuraminic acid, this is an organic acid. We are going to study them in metabolism. This is pyruvic acid, okay? And the remainder is a derivative of mannose. Mannose has hydroxyl group at carbon number 2 to the left, hydroxyl group at carbon number 3 to the left. And then amino group replaces hydroxyl group at carbon number 2. So this is mannosamine. Amino group added to mannose. Mannosamine with an extra organic acid and another acid, acetic acid. Acetic acid is added to the amino group. So this is N. N means the amino group, and this is capital N. Acetyl, this is acetic acid, which is added to the amino group. We get capital A. Neuraminic, the name of the organic acid is neuraminic, capital N. Acid, we get capital A. So we have to know the classification. This is amino sugar acid. Glucose, this is the ester. What is meaning of the ester? Ester bond is a chemical bond, covalent bond, formed between any alcohol and any acid. It can be inorganic acid. It can be inorganic alcohol, okay? So, ester bond, any bond, covalent bond, formed between acids and alcohol. This is the cyclic structure of glucose. I have discussed the cyclic structure of glucose in the first lecture. This is a cycle. At carbon number 1, there is hydroxyl group. Hydroxyl group at carbon number 1. If we add phosphoric acid, P means phosphoric acid, and around which a circle, this is an abbreviation of the formula of phosphoric acid. So this is inorganic acid attached to an alcohol. It makes an ester bond. This is glucose 1-phosphate. Why it is beta? I discussed beta and alpha in lecture number 1. Beta because the hydroxyl group at carbon number 1 in carbon number 1 is oriented to the left. The second one is glucose 6-phosphate. If you look, carbon number 6 of glucose, there is an alcohol group and a bond between alcoholic group and phosphoric acid. At carbon number 6, this is glucose 6-phosphate, and this is an ester, monosaccharide ester with phosphate. If you look at this formula, this is galactose. Galactose has hydroxyl group at carbon number 3 to the left, carbon number 4 to the left, but there is sulfuric acid, sulfate. Attached to carbon number 3, it enters in the structure of glycolipids, okay? Glycosides. Glycosides are molecules which is formed by a condensation of the anomeric carbon. This is carbon number 1. Anomeric carbon is the carbon which contains aldehyde or ketone group, okay? In aldehyde, in aldosis, it is carbon number 1. In ketosis, the ketone group in carbon number 2. So the anomeric carbon contains the carbonyl group. The carbonyl group is either aldehyde at carbon number 1 or ketone at carbon number 2. Condensation means covalent bond formed between two molecules by removal of water. This is the definition of condensation. It is a covalent bond formed between two molecules by removal of water. Condensation between any sugar with another sugar. Sugar is termed glycon. Sugar in organic chemistry is termed glycon. Or condensation between any sugar and non-sugar. So the compound is termed aglycon. A means no in French language. Aglycon, in French. Aglycon means not carbohydrate. So it is a chemical bond formed between sugar and another sugar, such as in disaccharides, or chemical bond, covalent bond formed between sugar, monosaccharide, and non-sugar, aglycon. The examples, glycoside, we are going to study this drug. It's called digitalis. It is used in heart failure. But the structure of digitalis, we don't need to know the structure of digitalis. Another form of glycoside, we are going to study them in nucleotides and nucleic acid. A condensation formed between ribose. Ribose is a monosaccharide, and non-sugar nitrogenous base. We are going to study them. Nitrogenous base may be adenine or guanine or thymine or cytosine or uracil. Of course, most probably you have studied these nitrogenous bases in your school, high school. It enters in the structure of DNA and RNA. Nitrogenous bases are organic molecule which contain cycle, which contain several nitrogen atoms, and this is not sugar. This is a glycan, not sugar, not carbohydrate. Condensation between ribose and non-carbohydrate give us glycosides. We have finished monosaccharide. What you have to need to know, you must know the classification and examples under each classification, because in the exam you will be asked, you will be given a certain monosaccharide, you will be given a certain monosaccharide derivative, and they ask you, does it belong to which classification? Or the reverse type of the question, give me an example of monosaccharide sugar acid, or give me an example of amino sugar or amino sugar acid. So you have to know the classification by name, and you don't need to know the classification by formula, never. Disaccharides are classified into reducing disaccharides, non-reducing disaccharides. Disaccharides, as the name state, di means two. Two monosaccharides condensed together to form a glycosidic bond. That is the definition of glycosides. Two monosaccharides linked together by a covalent bond to give us glycoside, which is disaccharide. You have to know the example of reducing disaccharide: maltose, isomaltose, lactose. Why these are reducing? As we will see in the formula, maltose, isomaltose, and lactose has free aldehyde group. Aldehyde group has reducing power. Any sugar which contains free aldehyde or free ketone group, it is a reducing sugar. Why sucrose is non-reducing? Because if you look at the formula, sucrose does not contain free aldehyde or ketone group. What's meaning by free? Free means that the aldehyde or ketone group does not enter in the covalent bond. It is not involved in the structure of the covalent bond between the two monosaccharides, because these are disaccharides formed between two molecules of monosaccharides. If the aldehyde group or the ketone group enter in the covalent bond, so it is not free and the sugar is non-reducing. If the aldehyde or ketone group does not enter in the covalent bond, so the aldehyde group is free and the sugar is reducing. Okay, you don't need the formula, so I'm going to go rapidly around the formula. Because this is the cyclic structure of lactose, you have to know that lactose is formed between galactose and glucose. Lactose is a disaccharide. It contains galactose and glucose. This is the covalent bond. If you look, this is the covalent bond between the two circles. You find two cycles: the cyclic structure of galactose, the cyclic structure of glucose. But there is an aldehyde group. This is aldehyde group in the cyclic structure free. Aldehyde group, so lactose is reducing, because the aldehyde group that is found on the right side of the figure, this is a free aldehyde group of glucose, does not enter in the covalent bond. The covalent bond, the bond which connect the two circles, because we have cyclic structure. If you look at sucrose, sucrose is formed between glucose and fructose, okay? There is no free aldehyde group. This is not an aldehyde group. This is an alcoholic group. Alcoholic group does not have reducing power, but aldehyde have reducing power, okay? This is a covalent bond which connect the two cyclic structure. This is a covalent bond. The covalent bond which connect glucose and fructose. Of course, you have to know and to study perfectly what are the hydrolytic products. If we hydrolyze lactose, we will get galactose and glucose. You have to study them. If we hydrolyze sucrose, you will get glucose and fructose, okay? This is maltose. Maltose has free aldehyde. This is an aldehyde group. Anyone want to ask me, aldehyde is CHO? This is not a CHO. This is actually an aldehyde, but in the cyclic structure, it looks like that, HOH. if you revise the formula of the cyclic structure, the first carbon is converted into HOH, and this is not an alcoholic, okay? Maltose is formed between two molecules of glucose, between carbon number 1 and carbon number 4, okay? This is carbon number 1, this is carbon number 2, this is carbon number 3, this is carbon number 4, this is carbon number 5, this is carbon number 6, okay? If we count in the opposite direction, this is carbon number 1 in the other cycle. So the bond between carbon number 1 and carbon number 4, it is an alpha 1,4 bond between carbon number 1, carbon number 4, which connect the two cyclic structure. Isomaltose between carbon number 1, carbon number 6. Of course, you don't need to know the formula, but you have to know that isomaltose, there is a covalent bond between carbon number 1 of the first glucose, carbon number 6 of the second glucose, because both maltose and isomaltose have two molecules of glucose connected together, linked together by glycosidic bond or covalent bond. Oligosaccharides. We have finished disaccharides. You have to know examples of the reducing disaccharides. 3 examples. You have to know examples of non-reducing disaccharide, 1 example. And to have to know the hydrolysis. If we hydrolyze lactose, we get which type of monosaccharide. If we hydrolyze sucrose, we get which type of monosaccharide. And you have to know the cause, why sucrose is non-reducing and why maltose is reducing, okay? You have to know several aspects in disaccharide. Examples, the cause of the reducing power or non-reducing power, and the hydrolysis. If we hydrolyze any type of disaccharide, we will get 2 molecules of monosaccharide, which are these 2 molecules, okay? Oligo is not important in our diet because they are non-digestible. Why we study these oligo? Oligo means little يعني little number, from 3 to 10 monosaccharide units connected together by glycosidic bond or covalent bond. Oligo means a small number of monosaccharides linked together in a chain by covalent bond. Why we study them? They are not important in our diet. They do not give us energy. Actually, they are sensitized in our body, enter in the structure of antigen, such as the blood group antigen, enter in the structure of glycolipids, enter in the structure of glycoproteins of the cell membrane, and we are going to study all these molecules and their biological function. Of course, not every molecule in our body give us energy. For example, hormones does not give us energy. We do not oxidize hormone to get energy, but hormones has very important biological function, essential for life. Blood group antigen has important function. Immunoglobulin has important biological function. Cell membrane has essential biological function for the vitality of the cell, or the cell is going to die. Okay? So oligosaccharide play physiological and biological function but does not give us energy. The last thing is polysaccharide, and I write beside them they are non-reducing. Why they are non-reducing? Because they don't have free aldehyde group, they don't have free ketone group. Both aldehyde and ketone are called carbonyl groups. So there is no reducing power in polysaccharides. The definition of polysaccharides: polysaccharides contain more than 10. Actually, they are not 11 or 12. Actually, they are several hundreds. There may be 500, 1,000 monosaccharide linked together, and the structure is highly complicated. A polysaccharide contain several hundred, several thousand. But the definition say more than 10 molecules of monosaccharide is defined as polysaccharide. This lecture we are going to begin polysaccharide, but the remainder of polysaccharide will be in the 3rd lecture. Polysaccharides are classified into homopolysaccharide, heteropolysaccharide. What are HOMO? HOMO is formed by the same type of sugar. For example, if we get several hundreds molecules of glucose and we link them together in a chain to make a chain formed by the same type of sugar, which is glucose, so it is termed homopolysaccharides. On the other hand, if I get glucose and link it to fructose, and fructose link it to mannose, and mannose link it to glucosamine or any other monosaccharide derivative, so I link different types of sugars to the structure is called heteropolysaccharides. Today we will only discuss homopolysaccharides. The first are glucan. As the name said, glucan from glucose. We are going to study in glucan, starch, glycogen, dextrans, cellulose. Okay? Fructans are homopolysaccharide formed of fructose units. Several hundred molecules of fructose linked together to give us fructans. You have to know only the example of inulin. Inulin has functions in our body. We are going to study them in physiology. These are derived from plants, okay? Glucans. We are going to begin with starch. Starch is the storage form of carbohydrate in the plant cell. If you look at the plant cell under the microscope, under the ordinary light microscope, you are going to see the starch as granules formed of several layers. Any plant which contains chlorophyll, green plant, sensitizes starch. As we know, the green plant takes the light from the sun and takes some nutrients from the soil, nitrogenous compound from the soil, and sensitizes starch as the storage form of carbohydrates. Any plant cell, even the green leaves which we eat in our diet, contain starch. So starch is a polysaccharide formed of several hundred molecules of glucose linked together by a covalent bond. It is the major storage form of carbohydrate in plant cell. What is glycogen? It is similar to starch. There are certain differences, minor differences. The storage form of carbohydrate in animal cell, in our body, we store carbohydrate in the form of glycogen in the animal cell because human belong to animal kingdom, human being. Okay? So if we get several hundred molecules of glucose and link them together in the animal cell, it is termed glycogen. If we make hydrolysis to either starch or glycogen, and we are going to make hydrolysis in the laboratory work, so these are dextrans. Dextrans are the product of hydrolysis of either starch or glycogen, but usually it is the hydrolysis of starch by salivary amylase. This is the enzyme secreted by our saliva in the mouth, or the pancreatic enzyme termed pancreatic amylase. The last one is cellulose. Cellulose is a polysaccharide formed in the cell wall of plant cell. If we eat any plant cell, green leaves or potatoes or any type of plants, okay, the cell wall, similar to the cell membrane of the animal cell, the cell wall of the plant, if you touch it, it is much harder than the cell wall of our body. Our body is soft. Cellulose is hard. It is non-digestible, but it's very important in our diet. Although it is not digestible, it's non-digestible, does not give us energy, but it helps to stimulate our intestinal motion, small intestine peristalsis, large intestine peristalsis, to help the motion of our intestine to prevent constipation. And there are evidence that cellulose prevent cancer of the colon and cancer of the GIT, of the gastrointestinal tract. We are going to study the structure of the cellulose in the next lecture. Although they are non-digestible, but they are very, very important in our body, and you have to eat plant diet. You have to eat the cell wall of the plant of the green leaves because this stimulates intestinal peristalsis and prevents constipation and prevent any cancer of the gastrointestinal tract. Okay, this is the beginning of the homopolysaccharide, as you see, starch is formed of amylose. Amylose is formed of several hundred molecules of glucose. This cycle is glucose. This cycle is glucose. This cycle is glucose, and etc. We have several hundred molecules of glucose linked together in a straight chain. This structure is termed amylose. Okay? In another structure termed amylopectin, there is a branch. If this is a chain formed of, while التعليم ما بيكتبش, chain, and the chain is branched, give us branch, and you will go to another chain, another branch. So this is branch. I make like an arrow. Okay? So if we have several branches linked together by side branch, this is amylopectin. So starch is formed of amylose, which are glucose molecule formed between carbon number 4, the chemical bond, the covalent bond of the first glucose molecule, and carbon number 1 of the second glucose molecule. Okay? So all the bonds are formed between carbon number 4 of the glucose molecule on the right side, carbon number 1 of the glucose molecule on the left side. So the chemical bond is alpha 1,4 glycosidic linkage or glucosidic link. But in this branch, this is carbon number 1 and this is carbon number 6. So the branch point is a glycosidic bond between carbon number 1 of the glucose in the upper chain and carbon number 6 of the glucose molecule in the lower chain, and all glucose molecules are colored by the rose color, the light pink color. Okay? To make it easy, we are going to study in details polysaccharide in the third lecture. Thank you for your attendance and your patience, and this is a joke. Okay? Thank you.